That therapeutic piece of RNA, that's what encodes for that broken gene that's in the blood stem cell in the case of ALD. Here you see it packaged, and boom, right there, that's your virus, that's our Trojan horse. That's the thing, it's actually an HIV-derived virus that now has been hacked up and is produced, and now millions of those are sitting in this vial. Cool, but not that helpful unless you have the other half, which is you need the blood stem cell from the patient, from Ethan. And here what you're visualizing is you're going into the you're going into the marrow to pick out the blood stem cells. And you see these little red guys popping into the bloodstream? Those are the ones we capture. It's a fairly standard procedure. We stick billions of those, or as many as we can, into a bag. Now, next step, virus meets blood stem cell in the bag outside the body. So virus never goes in the body. What it then knows to do is to infect the cells. Infect it as many times as we can, as many cells as we can in that bag. It goes into first the cytoplasm, then it releases the payload. It then knows to, and we're certainly taking some scientific liberties here to make it a little more simple, but it explodes there, right there. That's your money ship, the RNA. But for those of you who remember your biology class, we got to do some gymnastics to make sure that turns into DNA. Because we have here, up here it says revolutionary ideas. Well, watch carefully here, because as we finish this gymnastics, you're going to visualize the revolutionary idea. This gets moved and transported into the blood stem cells compartment, the nucleus, where the DNA exists. And right here, in a little magic, boom, it integrates itself into the DNA. Now, what you've just created is a gene-modified blood stem cell. That stem cell knows what to do. The cells know what to do. It starts producing your therapeutic gene. It starts producing a lot of them. You turn your red cell into blue cell in this picture here. Bottom line, those are modified. Those now know how to do what the problem was before. We put it back into the patient. So picture Ethan here. We put it back in. Now these cells seed your marrow, and they then start to produce daughter cells. All those daughter cells now know what to do. They have a functioning